Hello lovelies, in this video we're going to be looking at parts of respiration, glycolysis. This is going to be the first in a series of videos taking you through all of the different parts of respiration that you need to know.
Hi everyone. Okay, so we've done photosynthesis, so now we're going to start talking about respiration. So first of all, let's think about the importance of it and why do we need it. So respiration is a series of small chemical reactions which are controlled by enzymes. That's important because if we didn't have those small steps and we just let rip and sort of released all that energy in one go, then that would be disastrous for kind of the cells and there would be lots of heat released and it would be very dangerous. They release energy in the form of heat, which they do. In some places the energy is wasted as heat, but also through they produce, so they sort of trap and contain that energy by producing ATP. And ATP is our short-term storage molecule, sometimes called energy currency, because it's what is used to store and release energy in energy requiring reactions and then also energy producing reactions like respiration. It's a process that's carried out in some form in all living organisms. So we're going to talk about kind of anaerobic respiration as well. We know that some prokaryotes do this. There are some prokaryotes that have other forms of energy production, thinking about ones that use methane instead of glucose and things like that. So, but there's sort of basic threads that run through all forms, and it's this way of making ATP. Although photosynthesis does produce some ATP, as we looked at, it's not enough to sustain all the processes in a plant cell. So this is how this photosynthesis and respiration kind of relationship has been made. Okay, so let's just sort of look at how these two are interlinked. So our light energy comes from the sun. That obviously hits the chloroplast, and the chloroplast goes through photosynthesis using that light energy to produce glucose and oxygen, which we've looked at already. That glucose and oxygen can then move within the cell to the mitochondria. So it's in the cytoplasm, it will leave the chloroplast, remember oxygen will leave the chloroplast because it's a waste product, and the glucose will leave once it's gone through the Calvin cycle, and that will be in the cell, ready in the cytoplasm, ready to use. And then these will travel to the mitochondria, where aerobic respiration will take place. And that produces more carbon dioxide and water as waste products, which can obviously then be absorbed by the chloroplast again to do photosynthesis. And the mitochondria, where the aerobic respiration is happening, we're releasing energy in the form of ATP. So we're storing energy, small amounts of energy in those bonds of ATP. So remember, we can have a MP, which is monophosphate, a DP, which is diphosphate, and then we have a TP, which is triphosphate, and the A stands for adenine, which is one of those bases, the organic bases. So ATP is technically a nucleotide. So we need to remember why ATP is useful as our energy currency, as our energy releasing molecule, is because what it does is it's very easily made and very easily broken. So it releases small amounts of energy right where the energy requiring processes are. So, and it is able to be dissolved in the cytoplasm, and it can kind of get to where it's needed very quickly. And then you hydrolyze the ATP to ADP, and you've released that phosphate, but that release of that breaking of that bond releases a small amount of energy, which can then be used for energy requiring processes like active transport, anything like that. Let's have a quick overview of what we're going to go through when we talk about respiration. There are 4 main stages. So in this video, we're going to look at glycolysis. It happens in the cytoplasm. It doesn't happen in the mitochondria. I'll explain all in a second. It splits glucose and it creates a small amount of ATP. Now remember, the whole point of respiration is to produce as much ATP as possible. So at each stage there is normally some ATP made, but overall that's going to add up to a large amount and much larger than photosynthesis. So that's stage 1. Stage 2 is called the link reaction. It literally links glycolysis with the next stage, the Krebs cycle, and it's how we get the products of glycolysis into the mitochondria. It does not produce any ATP, but it happens in the mitochondria. The Krebs cycle is another cycle. It's going to be similar to the Calvin cycle that we've already learned. It sort of does a similar job. We're producing a lot of coenzymes, so NADH and FADH, so lots of reduced coenzymes which are going to carry electrons and hydrogen ions all the way to the ETC. Very, very similar to what we had previously in photosynthesis. So we're producing coenzymes that are reduced because they have the added H plus and electrons, which are going to go to the ETC, an electron transport chain, in order to produce ATP. And then lastly, we have oxidative phosphorylation. Now, we've spoken about what phosphorylation means. It means adding a phosphate. So we're making ATP by adding lots of phosphates to ADP, using ATP synthase in exactly the same way as we talked about in photosynthesis. But we're not talking about photophosphorylation here because there's no light. So we're talking about oxidative because we're going to be using oxygen, which is what the main reactant of respiration is. So it happens in the membranes of the mitochondria, similar to how the electron transport chain happened in the membranes of the chloroplast. And so it's going to be a very similar story. And that will be where we make the most, like the majority of our ATP from respiration. Okay, so we're going to have a look at stage 1 of respiration, which is known as glycolysis. The first stage of both aerobic and anaerobic respiration is glycolysis. They both share this stage, and we're going to look a little bit at anaerobic respiration at the end. It takes place in the cytoplasm of the cells, not the mitochondria, and that'll make sense in a second. It uses up glucose, and it produces a small amount of ATP, but this isn't obviously the main part of aerobic respiration because aerobic respiration is needed to produce a lot of ATP. Glycolysis literally means, if you break the word down, glucose or sugar splitting. Because remember, lysis, we've seen this word a lot. It means to break or to split. So we are literally, the process of glycolysis, the point of it is to split glucose. And we're going to split it into 2 molecules that we're then going to use in the next part of respiration. So just a reminder of a location of where this is all happening. So we've got our mitochondria. This is a plant cell, obviously. Plant cells do respiration, the same as animal cells also do respiration. And then we've got the cytoplasm here. So we know that aerobic respiration happens in the mitochondria. And I've just told you that glycolysis doesn't actually happen in the mitochondria, it happens in the cytoplasm. Now that makes sense because it can't be in the mitochondria if only aerobic respiration happens inside that organelle. So because this is the first stage, kind of the precursor stage to aerobic respiration and anaerobic respiration, it takes place in the cytoplasm. So realistically, when we say aerobic respiration happens in the mitochondria, what we mean is all of the stages after glycolysis, if there's enough oxygen. Glycolysis takes place in the cytoplasm, and that's because it contains all the enzymes that we need in order to do this process. And obviously, it's where the glucose will be as well. As the first part of respiration, glycolysis can be used to produce small amounts of ATP when there's no oxygen. So this is the process that's very useful. And if there is no oxygen, we go through from glycolysis into anaerobic respiration and we cycle round, and we'll talk about that at the end. It's not going to produce enough ATP for us to carry on surviving if we don't get enough oxygen at the end. But it's thought that because this produces a small amount of energy, which would have been suitable for something like a single cell or a prokaryote, it's thought to be an ancient form of respiration, kind of the short form, the kind of easy way of getting energy from a molecule that probably evolved and started off this process of us having aerobic respiration at some point. Some things to remember and some new information that we've just talked about that we've got to cover before we start. So we must remember oil rig. We've got to remember that oxidation and what that means. So oxidation is loss of hydrogen ions and electrons, and then reduction is gain of hydrogen ions and electrons. And all of the redox reactions that happen in this series of reactions that we're going to look at involve coenzymes. So we haven't used this term yet before, but we have seen coenzymes already. So coenzymes help enzymes to catalyze redox reactions. So they help them do their job of reducing or oxidizing molecules. They act as the donors or the acceptors of the hydrogen ions and the electrons. When something is reduced or oxidized, those hydrogen ions and electrons have to go somewhere. So the coenzyme takes them, not the enzyme, and then they get reduced or oxidized, and then that's how we transport our hydrogen ions and electrons around inside, as these are often known as carriers. So they're sometimes called protons or hydrogen ion or just hydrogen carriers because they are carrying hydrogen ions. And we have learned about a coenzyme already. It's NADP, which is the hydrogen carrier that we've seen in photosynthesis. And we've got a very similar one here in respiration. So in respiration, what examples have we got? We've got NAD. So we've just dropped the P. I like to think of it, and the easiest way to think of it is that NADP has a P for photosynthesis. So you only add the P to NAD if you're doing photosynthesis. If you're doing respiration, there's no P, it's just NAD. And it has the same role. It takes hydrogen ions and electrons and is going to take them to the electron transport chain. We have another one, FAD, again, similar to NAD, but doesn't contribute to ATP synthesis. We'll talk about this when we get to the electron transport chain. And we have coenzyme A. Coenzyme A is going to be carrying an acetyl group from the link reaction into the Krebs cycle. So it's something that carries an actual group of atoms rather than just hydrogen ions. And it's going to help us with our link reaction, getting our products from glycolysis into the Krebs cycle. So when we talk about NAD, FAD, and coenzyme A has it in there already, but they're all coenzymes. They're all going to be at some point reduced or carrying hydrogen ions and electrons to another part of the mitochondria in this case, or another part of the process of respiration. So the first stage of glycolysis, it's broken down to about 4 stages, is phosphorylation. So we are phosphorylating. Remember, that means adding a phosphate. So we are adding some phosphates to glucose. So that glucose can come from the blood or it can come from a breakdown of glycogen. We phosphorylate it twice. So there are times 2 phosphorylations here. And this activates it. So by adding a phosphate, remember, you activate a molecule or you can raise its energy level. So we need 2 lots of ATP to do this. And that means we're making 2 ADP as well. So that's thinking about what our products are going to be. Then we are making 2 ADP. And we make a molecule that is called hexose bisphosphate. You can also see this sometimes referred to as fructose 6-phosphate. So hexose bisphosphate is in biphosphate, 2 phosphates, 6 sugar molecule. If we're being specific about which 6 sugar molecule we've got, it's fructose. So fructose 6-phosphate or hexose bisphosphate, same thing in terms of what the molecule is. Choose one and go with it. Okay, so the second stage is we've got our hexose bisphosphate or our fructose 6-phosphate, and it splits. So it's unstable. It splits into 2 molecules of our old friend triose phosphate or GALP, or in this case we can also call it GP. So this is the molecule we've seen before in photosynthesis. We've seen this molecule as like a precursor to things like glucose and other organic molecules. So we're reversing it here. We're splitting it, and so we've got from 1 glucose molecule up here. So there's 1 glucose molecule here. We get 2 GPs. And so then from here on out, we're making 2 of everything. So everything happens the same to each molecule. And so once we've got that split into the 2 GPs, we then go into stage 3, which is oxidation. Remember from our oil rig, oxidation is loss. So each TP, GALP, GP, whichever you're going to call it, is then oxidized. So it's losing H+ ions and electrons, and they're removed and they're donated to NAD because that is the coenzyme that's here. And so they are going to form reduced NAD. You can use various forms. So there are lots of ways you can refer to reduced NAD. We will keep it the same as we have done and just stick with reduced NAD because it's easier, but you may see it referred to as some other options. So what's actually happening here is we've got NAD, which is slightly positive, and we are adding 2 H+ ions and 2 electrons, and that is giving us, therefore, NADH plus H+. So you can see it referred to as NADH plus H+. Sometimes this is also abbreviated to NADH2. So you can see it as that. I would say if we stick with it, I would think the best thing to do is to stick with calling it reduced NAD. And then we don't get into any of those pickles about whether we're saying pluses or not pluses. So far, we have used 2 ATP, and we are also producing 2 reduced NAD so far. Okay, so we're at our last stage now. So we have taken our GP, TP GALP, and it has been oxidized. So we now need to convert it to pyruvate. That is this stage here. So from here to here, we are converting our GP or our oxidized GP into pyruvate. I've blurred out the names of all the molecules here because we don't need to know these. You do need to know hexose bisphosphate or fructose 6-phosphate. You do need to know glyceraldehyde 3-phosphate, which is GP, or you can call it triose phosphate or GALP. And we do need to know pyruvate, but we don't, this conversion stage, we don't need to know. We are just needing to know that we are going to use, we're going to convert it. And that conversion, using enzymes, again, that we don't need to know the name of, but that conversion produces 2 ATP per molecule. So we are able to release some energy as we convert these molecules, and that releasing of energy actually then is used to phosphorylate ADP to form ATP. And so this phosphorylation of ADP to ATP is known as substrate level phosphorylation. It's happening right next to where the reaction is taking place. So that's why it's known as that, not like photophosphorylation or oxidative phosphorylation, which we're going to come on to later. So in total, we actually make 4 ATP from this part, just this conversion stage. However, so we've got 4 ATP being made. However, the net gain, which just means the overall gain from the whole of this reaction from glycolysis, is only 2 ATP because 2 were used at the start up here, if you remember, to phosphorylate the glucose at the beginning. So because we used 2, we made 4. Overall, we make 2 as a net gain. Okay, so that was a lot, but we can summarize it quite short. So 1. Glucose is phosphorylated to hexose bisphosphate using 2 ATP. The hexose bisphosphate splits to produce 2 molecules of TP, triose phosphate or GP. The triose phosphate or GP is then oxidized to form pyruvate, also forming reduced NAD in the process, and this releases energy which produces 4 ATP. That is a very basic, quick summary, and it is basically what we've used, like what has happened in that long sequence. There are some little details that we may need to add in, but if you are asked to kind of summarize glycolysis or explain what happens, this is what happens. So let's have a think about what we're producing then. What are the products of this reaction? Per glucose, so to start with 1 glucose molecule, what do we get out at the end? Well, we get 2 reduced NAD. So we make 2 reduced NAD. So remember, there's no P. We're not talking about photosynthesis here. They diffuse, because obviously remember this is happening in the cytoplasm. They diffuse into the mitochondria, and they're carrying, remember, the H+ ions and the electrons, and they're taking them to the electron transport chain. So we make 2 ATP net. So overall, we make 2 ATP that are not going to be used at the start of glycolysis. So these are used for energy requiring processes that are happening nearby. You can use them for, the cell can use them for various things. What is probably likely that they're used for is that we do need to actively transport pyruvate into the mitochondria for the next stage, which is part of the link reaction. So it can be used for that. Okay, so we also make 2 pyruvate, which is the whole point of glycolysis. Remember, we're splitting glucose. We literally split it into 2 molecules of pyruvate, and the pyruvate is what we need to carry out the next stage of aerobic respiration, which is the link reaction. So it's actively taken up by the mitochondria into there to go through the link reaction in order for us to carry on with aerobic respiration. But that only happens if there is enough oxygen. If there isn't enough oxygen for that to take place, then the pyruvate stays in the cytoplasm and it gets used in either alcohol fermentation or lactate fermentation, depending on which type of organism we're in, either animals and plants or bacteria. And So it's dependent on whether there is oxygen, whether this goes through to the link reaction or it stays in the cytoplasm for anaerobic respiration. So we're going to take a small detour just to quickly talk about anaerobic respiration because it's not very complicated once you've done glycolysis. Alcoholic fermentation is what it's called in plants, yeast, and in some bacteria, not all. They use the reduced NAD to convert pyruvate into ethanol, and this process also produces carbon dioxide. So pyruvate first gets decarboxylated, so carbon dioxide gets removed to form ethanal, and then we use our reduced NAD to oxidize the ethanol into ethanol. So we produce a carbon dioxide and we produce NAD. That's important, and we'll come to why in a second. In animal cells, it's called lactate fermentation because we're making lactate or lactic acid. So again, we're using our reduced NAD that we've produced at the end of glycolysis to convert our pyruvate to lactate, which otherwise known as lactic acid. So why is this important? Why does this link back to glycolysis and why do these organisms do anaerobic respiration in this way? Well, both of these fermentation processes allow the regeneration of NAD. So we're using the reduced NAD and therefore we're creating the NAD. So what we do then is we allow glycolysis to continue because if we've got more NAD and we can go back and do the same process again, we can do glycolysis to make more pyruvate, and then we can go through the same conversion stage to get our fermentation. So it allows small amounts of ATP to be continuously made through glycolysis, and it allows it to continue happening so that organisms can survive for a short time without oxygen, because the energy is going to be able to be used to do some basic fundamental processes. So often there's questions about how does fermentation or how does anaerobic respiration allow small amounts of ATP to be produced, or why is it important that NAD is regenerated in fermentation? And that's the kind of answer they're looking for. Linking a little bit back to GCSE here, how do we get back from lactate or lactic acid? So in animal cells, after some time, then we can reoxidize that lactate back to pyruvate, but we have to use oxygen to do that. And that oxygen is what we say the oxygen debt is. So the amount of oxygen we used to say needed to convert lactate back into, or to break down the lactic acid, that's what we're saying here. What we actually mean is we're saying we're oxidizing the lactate back to pyruvate, then that can go back to the start of the link reaction, and therefore we can continue with our respiration reactions. Some lactate can also be converted to glycogen and then stored in the liver and muscle cells. So that's another way we can kind of get rid of that lactic acid and store it as something that can be used. So glycogen can obviously be used to be broken down to resupply the cells with glucose for respiration as well. That was it. That's a summary of the first stage of respiration, glycolysis, including the little bit about anaerobic respiration. Hopefully that was helpful. We're going to go straight into the link reaction in the next video.
Ouch! This is why in some videos I have unexplained scratches.